Remdesivir is the first antiviral medicine to be effective in fighting SARS CoV-2, the virus that causes COVID-19, and represents our first hope in treating this terrible disease. Remdesivir is an investigational drug and has received emergency use authorization for the treatment of hospitalized patients with severe COVID-19 disease. Remdesivir, originally developed for use against Ebola virus, was discovered by scientists at Gilead Sciences in collaboration with USAMRIID and the CDC. In this video, we're going to probe remdesivir's mechanism of action and understand how it inhibits viral replication at the molecular level. The virus invades the host cell and hijacks the cellular machinery to make more copies of itself. The SARS CoV-2 genes are encoded in a strand of RNA, which are read by the host cell to synthesize viral proteins. One of these proteins is an enzyme, RNA-dependent RNA polymerase, which churns out more copies of the viral RNA necessary to make more virus particles. At a high level, remdesivir works by inhibiting this enzyme and shutting down the viral replication process. Let's take a closer look at how this works. Remdesivir mimics adenosine monophosphate, an endogenous or naturally occurring nucleotide. Remdesivir's structure is different in a few critical ways. First, we see that there is a cyanosubstituent on remdesivir that is not present in adenosine monophosphate. This is critical to the mechanism, and we'll talk more about the cyano group in a minute. Second, we see that there are additional substituents on the phosphate group. It's actually remdesivir monophosphate that we want to get into the cell to do its job. But remdesivir monophosphate cannot pass through cell membranes. The phosphate group carries a negative charge and is too polar. The phosphate group has to be masked, converting it to a neutral phosphoramidate ester, which is much less polar. Then it can pass through cell membranes unimpeded. Inside the cell, an esterase cleaves the ester group, leaving a phosphoramide. This is a substrate for phosphoramidase, leaving a monophosphate ester. The phosphate group prevents diffusion out of the cell membrane and traps the intermediate inside the cell. The monophosphate is converted by host cell kinases to the triphosphate, which then acts on viral RNA polymerase to shut it down. Remdesivir is a prodrug, that is, it is a precursor for the active form of the drug, remdesivir triphosphate. A minute ago, we were depicting RNA-dependent RNA polymerase as a spinning wheel, cranking out viral RNA, and here we're showing it as a space-filling model. This enzyme works by using viral RNA as a template to make additional copies of viral RNA. It's a highly efficient enzyme that can add 100 nucleotide bases per second. Each nucleotide base that is added to the growing chain matches its complementary base on the template strand. The enzyme will use remdesivir triphosphate instead of adenosine triphosphate and incorporate remdesivir into the growing chain, which stops the process. The cyano group bumps up against the protein, causing an unfavorable interaction, which distorts the position of the RNA chain and shuts down the enzyme machinery. The enzyme adds a couple more bases, but then it stops, preventing the replication of the virus. Remdesivir actually has greater affinity for viral RNA polymerase than it has for mammalian RNA polymerase, thus increasing its antiviral activity and minimizing side effects. If you would like more information, here are two open access articles provided by the American Chemical Society discussing the discovery and clinical development of remdesivir. This and other articles are linked in the description of the video. To all the scientists who discovered remdesivir, great work. Your efforts to combat this disease are much appreciated. Thank you for watching, and please click the subscribe button.